Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
Welcome to the Deep Dive, where we really get into the nitty-gritty of some amazing science. And today, we're looking at the secret language inside our cells specifically, these tiny DNA switches that control how blood develops. But here's the thing that grabbed me right away, the real, counterintuitive bit. These switches, enhancers, they don't just turn genes on or off. Not that simple. This deep dive, based on a recent Cell paper from 2025, shows a single switch can sometimes activate and sometimes repress, or even, flip its role depending on what's around it. Kind of turns our understanding upside down regarding cell precision. Okay, so let's unpack this a bit. What exactly are these enhancers we're talking about? Well, you can think of them as short stretches of DNA, kind of like molecular light switches for our genes. They get bound by these special proteins, transcription factors, or TFs. And these TFs are absolutely crucial for turning a basic stem cell into, well, any of the 200-plus specialized cells in our body. Now, for years, we've known where loads of these enhancers are. Big projects mapped hundreds of thousands. But the huge problem, the real enigma, was why. Why does one specific little piece of DNA, say 150 base pairs, turn on a gene in one cell, like a macrophage, but stay totally silent in another cell right next door, like an eosinophil? It's like having a blueprint of all the switches in a giant house, but no clue which switch does what light, or why some switches work differently in the kitchen versus the bedroom. Yeah, this whole grammar challenge, figuring out the rules, like the order of binding sites, the spacing, orientation, all that stuff, it's been a massive hurdle. It really slowed things down in key areas, like reprogramming blood stem cells or engineering safer gene therapies. The old ways of figuring this out, just way too slow. And even fancy machine learning kind of struggled with the complexity, the tangled patterns you find in nature. So the scientific community was really crying out for some fundamental rules, almost like first principles. Rules to explain and predict how this enhancer syntax could create such specific instructions, especially during blood development, the hematopoiesis.
What's truly exciting here, though, is that this wasn't just an academic exercise just to understand basic biology. Not at all. Cracking this code, it held the promise of not just deep insights into gene regulation, but also really practical ways to build bespoke, custom-designed enhancers, which could lead to revolutionary gene and cell therapies. The potential impact on medicine is just huge.
Right, which brings us to the core dilemma these researchers were tackling. How do cells take these broad, kind of overlapping signals from TFs in stem cells and somehow translate them into sharp, clear on or off instructions for specialized cells? Like, why are stem cell genes often shut down even when the TFs that should activate them are right there and plentiful? Trying to figure this out by just looking at natural enhancers was messy, like trying to learn grammar from some dense, complex poetry. So they took this really clever bottom-up approach. They decided, let's just build them. Thousands of synthetic enhancers from scratch, where they can control every single detail of the DNA architecture systematically. And the design itself was, well, pretty ingenious. They created this huge library, over 64,000 unique synthetic DNA sequences, each one about 150 base pairs long. And they meticulously varied everything, the number of TF binding sites, their orientation, the spacing between them, for 18 key transcription factors known to be involved in blood development. Then came the really crucial part of the experiment. They put these synthetic switches into actual primary human blood stem cells, using a really cutting-edge CRISPR technique for precise integration. And then they let the cells mature, differentiate. And they tracked the activity of every single synthetic enhancer across 7 different types of mature blood cells, neutrophils, monocytes, the whole range.
Yeah, the scale and the precision here were just remarkable. It's a multiplexed approach. They ended up generating over 400 million data points, measurements of enhancer activity linked to specific cell states. Think about that for a second. It let them directly connect a specific DNA sequence they designed to its actual function, its activity inside a living cell as it differentiates. And crucially, in its proper genetic context, its native chromatin. It's like they created this perfect little lab inside the cell where they could just tweak one bit of grammar and see exactly what happened.
Okay, so they built all these switches, tested them meticulously. What was the first big surprise? What jumped out from all that data? Well, the initial analysis showed this really key difference. When they looked at enhancers that only had binding sites for one type of TF, these tended to give a more gradual response, maybe turning things on kind of broadly across many cell types or just staying off mostly. They didn't really give that sharp cell-specific on or off signal. But, and this is where it gets really cool, when they looked at enhancers with pairs of TF binding sites, oh, something totally different happened. These pairs created really precise, almost binary patterns, clearly on in one lineage, clearly off in another, or even actively repressed in a third.
Right. So what that really tells us is, while maybe one TF sends out a general let's get activated signal, it's the combination, the pair working together, that acts as the really fine-tuned specific switch for a particular cell state. These pairs are sophisticated. They can take those kind of fuzzy, overlapping TF signals present in the cell and translate them into very definite go or no-go instructions for differentiation. It's how you get that clean separation between different blood cell type, that hierarchy.
Okay, that makes sense for the pairs. But they dug deeper, right? Even into the behavior of the single TFs. And they found some weird stuff there, too. Some TFs, yeah, they consistently activated or repressed. Predictable. But others showed this thing they called cell-state-dependent duality, which means the exact same TF could turn a gene on in one cell type and then turn it off, repress it, in a different cell type, which points to like really complex roles depending on the context. But even stranger, maybe, was this occupancy-dependent duality. For certain TFs like CREB1, ELK1, SB1, just adding more binding sites or making them stronger didn't always mean more gene activity. In fact, sometimes an intermediate number of sites gave the most activity. Wow. But if you added too many sites, the activity actually went down or even flipped to repression. It's like a dim switch, right? But if you turn it all the way up, the light range just goes out. A really weird, counterintuitive dimmer switch.
Exactly. And this raises a fascinating question. How does a single TF manage that? It suggests that there's a pretty sophisticated mechanism at play. Maybe the TF activates one part of the process, but beyond a certain point, maybe when it's too crowded on the DNA, a repressive function kicks in. It's not just about quantity. This explains beautifully how even one TF in a steady environment can still create a sharp on-off pattern. It acts like a filter, only letting the just right level of TF activity through. It's about control, not just raw power.
Okay, wow. So single TFs can be dual. Pairs create binary switches. What about how the pairs interact? Because that seems like the next level of complexity. And this might be the most groundbreaking bit. They found this duality was pretty common for pairs too. But the absolute shocker was combinatorial duality. Get this. You take two TFs that on their own are activators. They turn genes on. But when you put their binding sites together in an enhancer, in certain cell types, they suddenly become strong repressors. They shut the gene down. It's like a negative synergy. 1 + 1 = -1 somehow. And crucially, the switch to repression wasn't just random. It really depended on the internal grammar of that enhancer sequence. For some specific pairs like FLE1, SPE1, or SEPA2, this repression effect was strongest when their binding sites were packed really close together.
And if we connect this to the bigger picture, they even used these sophisticated structural prediction models like AlphaFold 3. And it didn't look like simple physical blocking, one TF getting in the way of the other. That wasn't the explanation. Instead, the model suggested actual protein interactions, like maybe FLE1 could bind directly to SLE1 and physically change B1's shape or function, flipping it from an activator into a repressor in that context, which led them to this really profound idea. These enhancers are essentially sensing the ratio of different TFs in the cell. For example, that FLE1 SPE1 enhancer, it was repressive when FLE1 and SPE1 levels were about equal or FLE1 was higher. It only became active when the balance was just right, maybe skewed towards SPE1. So the enhancer can read these subtle quantitative differences in TF levels and turn them into sharp, decisive on signals, but only in the right cell state. It's incredibly elegant.
Okay, so these rules they found with the synthetic enhancers, the duality, the pairs, the ratio, sensing, how well do they actually apply to, real DNA in our cells? And what can we do with this knowledge? They tested that, right? Looked at natural enhancers. Yeah, they checked if these rules held up with actual genomic enhancers and even more complex synthetic sequences they build later. And the really exciting part is, yes, the design principles they figured out from their simple synthetic pairs, they were highly relevant. The activity patterns they saw in the more complex real-world sequences matched the rules they'd uncovered, which strongly suggests they've really cracked a fundamental part of the regulatory code, which then makes you wonder, okay, if you understand the rules, can you use them to design? Could they actually build, like designer enhancers from scratch, tell the enhancer exactly which cell types to be active in? So they challenged their models. They set up 64 different design tasks like, okay, model, Build me an enhancer that's on in these 2 blood cell types, but OF in these other 4. And the answer was a definite yes, using their model to guide the design process. They could automatically generate synthetic enhancers that did what they wanted with pretty high success rates. Around 75% for activation tasks, over 60% for inactivity tasks.
And what's so fascinating here is this isn't just theoretical anymore. It's a practical blueprint for engineering biology. This ability to design enhancers de novo from scratch, it means we could potentially write the precise genetic instructions needed. Tate turns specific genes on only in the exact cells we want to target. Think about gene therapy. Imagine how much safer and more effective it could be if the therapeutic gene only switches on in the diseased cells and stays completely off everywhere else. That's the potential here.
Now, for a really important comparison, they took all these findings from healthy primary human blood cells and compared them to what happens in the common leukemia cell line, K562. And the differences were, well, pretty stark, really telling. In the leukemia cells, many of those transcription factors seem to have lost their repressive ability. Remember those crucial neutralizing interactions in healthy cells where TFs could kind of cancel each other out? In the K562 leukemia cells, those interactions were often gone. Instead, you just saw broad activation. It's like the off switch or the dimmer function was just broken, stuck on. And why? Why that difference? Well, leukemia often involves changes, mutations that mess with the cell's overall gene regulation environment, the epigenetics. It often makes the cell generally more permissive for gene activation. Things get turned on too easily. Healthy blood cells, on the other hand, maintain this very delicate balance. They actually have fewer activating proteins compared to repressive ones in the nucleus, keeping things tightly controlled.
Exactly. And connecting this to the bigger picture, that difference is really profound. In normal, healthy blood development, those negative synergies where TFs effectively shut each other down or keep things quiet, they're absolutely vital. They make sure that stem cells keep genes related to differentiation properly repressed until the precise moment they're needed. It prevents chaos, prevents cells going down the wrong path too early. But that always-on tendency in leukemia, where lots of TF combinations just drive activation indiscriminately, that helps explain why cancer cells often inappropriately turn on genes from both stem cell programs and differentiation programs at the same time. It contributes directly to their uncontrolled growth and weird identity. It's a fundamental loss of that precise control.
You know, looking at this whole study really highlights the sheer brilliance and foresight involved. The authors saw this critical gap. We had maps of enhancers, sure, but we lacked a general rule book, a way to predict how a specific DNA sequence would actually behave, how it would restrict activity to just one cell type. And that lack of understanding was holding back basic science and practical applications like targeted gene therapy. Their bottom-up strategy, building thousands of these things, synthetically it seems like it was really the only way to uncover that hidden grammar, those causal rules.
Absolutely. The real power here is how they wove together 3 cutting-edge advances into this beautiful loop. First, the design. Using advanced AI to generate this massive systematic library of synthetic DNA, exploring the sequence space intelligently. Second, the validation, this incredible high-throughput system, putting those sequences into real human stem cells using CRISPR, and then tracking their activity across the whole blood differentiation tree, generating those hundreds of millions of measurements in the native context. That's key. And third, the feedback model, building an interpretable deep learning model from that data, not just a black box, but one that revealed fundamental rules. Rules like that graded duality, how TFs flip roles when clustered and cooperative gating, how spacing turns a general TF into a specific amplifier.
And they actually closed the loop, didn't they? They used their model to design completely new enhancers, aiming for ideal enhancers, specific just to eosinophils, for instance. And when they went back and tested those new designed enhancers, they got a 10-fold increase in cell type specificity compared to the best natural ones they could find. That's not just tweaking things. That's a massive leap, a truly monumental step forward.
It really is. This work goes way beyond just mapping what's already there. It creates, as you said, a kind of semi-axiomatic rulebook for actually designing enhancers. And crucially, it's backed by real predictive power. It fundamentally challenges that simpler idea that a TF is just an on switch or an off switch. It's so much more nuanced. And thinking about technology and applications, they've built this scalable platform. And an interpretable model you could potentially extend to other tissues, other cell lineages. It really opens the door for for rational design, safer cell therapies like CRT cells that only activate in the right immune context, or maybe even subtly redirecting cell fate by editing the grammar, the motif density in existing enhancers. If we connect this to the really big picture, this research feels like it's putting us right on the cusp of a paradigm shift. If these grammatical rules turn out to be generalizable, applicable across other cell types, then synthetic regulatory DNA, the control elements, could become as precisely engineerable as the protein coding sequences we've gotten so good at manipulating. Just imagine the possibilities for truly programmable synthetic biology for gene therapy, actually writing the operating system for life.
Wow. This deep dive has really shown us the incredible and often, real counterintuitive precision baked into our gene regulation, discovering that single TFs can wear two hats, activator and repressor, and that pairs of activators can team up to become repressors, all based on subtle tweaks in DNA grammar. It's pretty mind-bending stuff. And like you said, this isn't just some academic curiosity. It plugs directly into understanding development, understanding diseases like cancer, and maybe most excitingly, shaping the future of medicine.
Yeah, for so long, the focus has been on reading and writing the code for proteins, the what. But this paper and work like it brings us so much closer to reading and writing the how and the when, the actual control code of the genome.
So just imagine that future for a moment. A future where we can write the precise genetic instructions for almost any cell type, where we can design therapies to switch genes on or off with pinpoint accuracy, only in the cells that need it. All because we've started to crack this incredibly intricate enhancer code. It really leaves you wondering, doesn't it, what other fundamental biological languages, what other layers of control are still out there waiting to be deciphered, and what revolutions in health and disease might they unlock when we do?